So thank you all for sending me your presentations. I've been stealing bits from them for this summary. I'd like to be able to see you all if I can. So can you see the title screen with all the talks on? Yeah, good. Yes, some nodding there. Yeah. So we had 1, 2, 3, 4, 5, 6, 9, 10 talks in this day 2 of this session, and on a variety of subjects. A lot of greenhouse gas stuff, which is great. A lot of processes involved in greenhouse gas topics, and then a couple more focused on DOC and re-wetting of reservoir sediments. So the first talk in the session, let's put it even in slideshow, was on the direct effects of elevated carbon dioxide concentrations, and looking at the direct effects on 3 different organisms, Daphnia, ostracods, and a rotifer species. So this was Lana's, Alana, I'm not sure. Hi. Nice, very nice presentation. And it was an experimental setup. So it was looking at what was the impact of these high concentrations on biota. Some nice background information and setting of the scene here in this slide. And then they took 3 different CO2 concentrations and looked at the effects of different clones of Daphnia actually on ostracods and on these rotifer species. And she found some very marked effects of very high concentrations of PCO2 on mortality of species. It's really not great for water fleas, but not really great for any of them at the very high concentrations. And then she found also some more chronic effects on kind of on abundances and reproduction rates and growth rates. So I've just summarized each talk with 2 or 3 slides to try and remind you of the topic. So I would now invite anyone here who has any questions to Lana to go ahead and make them. I stop sharing my screen so that we can see each other when we do that. So are there any questions to Lana?
Your screen is still shared.
Okay, sorry. Thank you, Tom.
I actually have a question to Lana.
Okay.
Hi, Lana. It was hello. It was a very nice presentation. I was just wondering, how did you calculate the survival probabilities? I mean, did you count the organisms by taking them out of the jars, or how did you do that? In terms of
so the setup was basically one individual in one jar, so we could fill them up very detailed, and so mortality was very easily scored by just the day that it died. And then we calculated the survival probability using survival analysis based on whether or not they survived the 24-day treatment or not. So that's what this is based on. Really an experiment on individual level, not population or community level, because we wanted to see specifically on these species what is happening, and you can only do that in this kind of really experimental laboratory setup. But it does give us some insight on what will happen.
I see. So how many individuals did you use, for example?
Let me try to remember. So we did the 3 species in one setup. We were a bit limited by the setup. So I think we had 13 replicas per species per treatment. So in total it was about 120 jars, I think.
Wow. Okay.
So yeah, I would have liked to have more, of course, because when they die, you lose a bit of data. But this was only one experiment. I did some follow-up experiments that is not included in this talk, but there we gained some more insights.
Okay, thank you.
Yeah, Hannah, I have to admit I didn't see your presentation, but now that I see it, I feel like I want to see it.
Alright.
So I was wondering, like the concentrations of CO2, what kind of, like how high is that relative, like what kind of emissions would you expect with these concentrations for reference, and what would be the impacts of these organisms dying for the lake ecology?
Mm-hmm. So yeah, I also had a question about that on the forum. I noticed that I didn't include my concentration, so I'm sorry about that. It was just slipped my mind. But actually the control condition was around 1500 ppm, based on the current supersaturation state of natural lakes that we found in literature, and mostly in these types of studies that is used. The high concentration was 25,000 and the highest was around 80,000 ppm, so it's quite high. But mainly the aim of the study was to do a proof of principle that there are, besides indirect effects, also direct effects. So that's why we chose to use these quite high levels. And the 80,000 is we also indicated as an extreme level that will only happen probably in some extreme cases, but not, and also temporarily, not in a constant level. But the 25,000 is actually not that extreme. If we look at the gradients or the levels that are found now, they already occur. And it has been confirmed that the CO2 levels in the natural systems will increase even more. So they are already supersaturated and they will increase even more. And this is in the range of things that can be expected. But it's really hard to point your finger on this is the level that will happen, because in the literature, many different groups use different levels, just because there is no one prediction and all the systems are also different. It's also more difficult in comparison to the ocean, for example, because their ocean acidification study is actually a similar type of thing that happens. But the ocean is a much more stable, well-buffered system, and the predictions can be better done. And in a freshwater system, you have many external factors actually influencing the CO2 level, as I have seen in other people's presentations, which was really interesting for me. I don't remember the second part of your question. Sorry, can you repeat it maybe?
Of course, the impact of these organisms dying for lake ecology.
Yeah. Well, actually, the main effect that we found was not mortality, only under these extreme levels. So I think if there is a temporarily massive CO2 level occurring and they do die, then I think a lot of other species will die also. So it's just will be detrimental to the entire system. Only the algae will like it, of course. They love CO2. But in this case, I think will be a bit more subtle. So I don't think that the mortality will be a big issue, but the changes in life history. For example, we found a large reproduction response so that they have less offspring, and this can influence the population dynamics. And we also saw that the ostracod was a bit more tolerant because they show some acclimation. I did not include it in the presentation, I think, but we found some acclimation. So they might adapt over time, while the Daphnia did not show this acclimation. So we expect a shift in community composition from maybe larger bodied species to smaller bodied cladostrons or ostracods or rotifers. But it's really hard to make predictions from this experiment alone. So we'll have to see.
Okay, thank you.
I was wondering if you measured oxygen at the same time, how oxygen responded to these different concentrations, and how you think that might be, and if it's a proof of concept, what's your idea for the next stage?
Mm-hmm. I think in this experiment we did not measure oxygen, but we did supply the jars where the CO2 was bubbled also with outside air, so it was sufficiently oxygenated. But the follow-up things that we did on this experiment is doing a kind of microcosm experiment. that I did recently, and there we did follow up both CO2 and dissolved oxygen levels and how it changes. And actually, we don't see a lot of difference between the oxygen levels because they are aerated. So that is probably, if you don't aerate them, I do think that they would not survive because we manipulate the CO2 level in such a way that it's an inside system. So if it's outside, they get the natural oxygen from outside. So it's difficult to say in this kind of setup, actually.
Great. Thanks for that. I think we'll move on to the next one, the next presentation. And this was by Tom Nyman. I think that's the correct pronunciation. I've been practicing. And it was on, took place in the beautiful Lake District of Denmark. And it was looking at how nitrogen fertilization and warming drive changes in methane oxidizing, in the methane oxidizing potential and capacity community in shallow lakes. And it used the experimental system from Aarhus University at Lemming, and it used this kind of the background was this very dodgy study from 2018 where they saw big changes in methane emission rates from the experiment at Lemming. But we have, so there's a big increase in bubble emissions at high nutrient and high temperature we found in this paper from 2018, and there's also been another paper from 2018 which found this in real lakes, looking at real systems. So warmer, more enriched tanks have a massive increase in abulative emission of methane, but we didn't have any analysis of the microbial community of the sediment, and so not much mechanistic understanding of the why these changes in emissions happen. So this was a great thing for Tom and the team to look at, and what they found was that warming and nitrogen increased methane oxidizer abundance. So this is reads, not any not actually potential oxidation rates, but this is the abundance of the microbial methane oxidizing bacteria. And then this increase in abundance also corresponded to a change in community type from type 2 to type 1 methane oxidizers at warmer temperatures. So we have this very nice plot here of temperature, so both seasonal temperature changes from these different periods of the year, week 1, week 8, and week 52, where the communities were sampled, but we also see the difference in temperature treatment as well. So I think that's the end of the summary of that presentation. So I'll stop sharing screen again, and I invite questions to Tom.
Hi, Tom. It was great presentation. I was just wondering, I saw that with increased temperature, you saw a shift from type 1 to type 2, right? And you concluded that this is due to the slower metabolism. Can you elaborate more on that? Why slower ones are gaining more advantage from this condition?
I read a paper about that, which was published in 2020 in Nature, Ecology, Evolution by Flux et al. And I think that what their hypothesis was that they modeled that, and they put made lots of different communities of two or three different microorganisms with different growth rates. And I think what they found was that slower growing organisms have a lower mortality rate than faster growing organisms. And that when temperature goes up, this mortality rate is more important for being able to compete with the other organisms. And then because the slower growing organisms have a lower death rate, they win the competition. I think that was how they thought it worked, and how they showed it worked. I could maybe I think you could find it if you look at if you search for this. I thought it was a very interesting paper to read. So and then put like slower growing microorganisms.
Okay, thank you very much.
You're welcome.
Nestor has a hand up, unless it's an old hand.
Hi, Tom.
Hi there.
Hi, Tom. How are you? Well, good morning. Excellent contribution and a very good combination of approach to understand the causality or the causal mechanism related to the observed pattern. In this sense, I have a small question. The community, the bacteria community that you analyze, only use nitrate as sources of oxidation, or can use another or other alternative oxidizing agent?
Thanks, Nestor, for your words and your question. So the methane oxidizers primarily use oxygen as an electron acceptor, and the ones that we focused on are all aerobic methane oxidizers, so they all use oxygen. There is also a group of methane oxidizers that uses nitrate as an electron acceptor, and they were also present, but at much lower abundance than the aerobic methane oxidizers, which use oxygen as their electron acceptor. And I think there are even other types of electron acceptors that can be used, but I would say that these are the most important. As far as I know, these are the most important too. And actually, most of methane oxidation happens, especially in shallow lakes. Most methane oxidation is aerobic, so with oxygen as electron acceptor. Does that answer your question?
Thank you. Thank you.
Okay.
Any more questions?
I'll ask a question.
Sure.
This is very interesting, and I'm not as familiar with the microbial side of things, but I did read the Zoo Nature Climate Change article from, I think it was 2020. Is that the one you were mentioning? And they were looking at the methanogens and the methanotrophs, and they found that the methanogens, I think, ramped up under warming, but the methanotrophs, their community was conserved. And I'm wondering if you can kind of tell based off of what you did and what they did, like why you may have seen differing results, if I'm getting both studies right.
So I haven't mentioned that paper in my presentation, but I have read it, and I think it's a really interesting paper. I think I don't know it in enough detail to really say what's different. But what I can say is that their setup is a little bit different. So we have a much longer term experiment. So I think that, for example, these shifts in community composition can take a long time to occur. And this experiment was running when we measured for 17 years, I think. And also, I think we have a higher resolution of measurements. So I know that they looked at methane oxidation, but they did it for a part of their mesocosms and not every time that they measured. And we took samples of every mesocosm every time, and then we looked at the microbial community. So maybe that's also partly explaining why we found a different result. And regarding methanogens, we actually just received data on that, so we'll also look at that and then compare. I'm really curious to see. I think their paper was mostly about methanogens, so I'm really curious to see if it compares, if it's similar. Yeah, but thanks.
We've got time for one more quick question on this one, if anyone wants to.
I have a question for you, Tom. Hi. I'm really fascinated about your work and how you work with this methane, and especially the fact that you say that that study found that higher temperature promotes methane evolution. So is it then actually a vicious cycle that with climate warming, more methane will be emitted from the water systems into the atmosphere and then warming again? So how do you think we can manage this, or what can we do to work against this emissions from the lakes to prevent it? Maybe something with your bacterial community that consume the methane. It's very interesting.
Yeah. I also, yeah, it's really interesting how you get this combination and then you get very, very high emissions. I think in terms of how can we, well, what's important to realize is that it's a combination of temperature and eutrophication that leads to the high emissions. I think it might be easier to do something about eutrophication than temperature, because that's really climate change. And regarding the methane oxidizers, I'm not sure if we can really influence them to have a different community composition. I don't know anything that you could add. If you could add something that changes the community composition, then maybe you can. But I wouldn't really know what. I think maybe it's important to focus on water quality and try to also prevent more. But yeah, there is a positive feedback loop here for sure.
Mm-hmm. Yeah, I agree. It's cool.
I would just add that I think there's a chronic shortage of data to really demonstrate this at a wider scale at the moment, and a lot of people are working on it to try and demonstrate it. This is an experimental demonstration of this, and there's been some comparative studies across landscapes that have shown it, and there's some theoretical, kind of half experimental, half theoretical work with metabolic theory showing that temperature increases methane emissions. But what we see again and again is that, as Tom said, eutrophication really ramps up methane emissions. So and that's something we can deal with much better on a shorter term. It's really still challenging, but stopping temperature increases. I mean, that horse has left the stable, right, for a good 50 to 100 years, or if we manage to do something. So it's really the eutrophication we need to get a handle on. Okay, I think we need to move on to the next presentation, and that was this one. Oh, wrong order. Share screen first. I think I shared the wrong one. Try again. So this was very my presentation. So I'm bound to say it's a very nice presentation, but it is a very, very nice study working on a single lake, which is Martin Sonnegaard also has a presentation on, which gives much more background detail, but I'll just talk about the fact that it's a 5 meter deep lake. It's still really a shallow lake because it's under 3 meters average depth if we use that definition, and that there was a strong stratification patterns over the summer period. So in June we had a very strong stratification pattern, and in August we had very nice strong stratification, and in between, when most Danes were on summer holiday, the weather was quite bad, so we didn't have stratification patterns. We had a lot of mixing, and you can see how that affects the dissolved oxygen towards the bottom of the lake. So we had no oxygen until we had mixing, then we had this period of variable mixing, and then stratification again. And we have a lot of measurements of greenhouse gases. We have these 4 transects of chambers which collect the bubbles, and we also measure dissolved concentration, very high frequency. Oh, and I've got all these, but basically showing that ebullition was much lower in periods where there was no stratification. So the stratification, and the longer the stratification, the greater the emission. And then these periods where it's a bit mixed, this is from June the 14th. So this is kind of bit of mixing, bit of stratification, bit of mixing. We have kind of intermediate emissions. What I think was really funny or interesting about this study is that I don't know if I mentioned it in the recording, was that I set we set these out, these chambers out to focus on the shallow areas of the lake, because that's where all the literature says the greatest bubble emissions were. So I didn't put, so there's only one set over here in the deep part, because I wanted to capture this depth gradient, which everything so far says bubbles come in shallower waters. But when the lake stratified, the bubble emissions were by far the greatest in the deepest waters. So be careful how much you believe of what you read. So when the stratification, so it's really, that's another indication of the fact that it was really anoxia driving these bubble emissions. So the shallow water stayed oxic longer during stratification as the point at which the lake was stratified moved up and down. I mean, we got anoxia up to 2 meters at certain points. So 3 meters of the lake was anoxic. So all these areas up to about, even only the shallowest bits would have had some oxygen in the at the sediment. And that's really the summary of the transects, or the averages of the transects. So you can see this point and this point are the main periods of extensive long stratification. That's not to mention the massive emissions when you had lake turnover and the bottom waters were exposed to the surface. Oops, went too far. A little preview of the next talk. So I'm open to questions.
Hi, Tom. I thought that was a great talk. I'm really excited about the interactions between mixing and some of these ecosystem properties. And I was thinking about the depth finding, right? Because you're right, everybody says that ebullition is happening in the shallower areas. And I was wondering if there was anything else that could explain the higher emissions. It seems like emissions may have been slightly higher on the eastern end where it's deeper. If you've thought about like any alternative explanations, the oxygen one maybe makes sense. I didn't realize that the shallow areas were oxygenated. But I wonder if you could elaborate a little bit on that.
Sure. Well, it's a work in progress. So I actually got an extension from Coca and uploaded it very late one night because we've really just worked these data up. So I haven't had the time to think about it in enormous detail. But what really struck me was that it's the deepest points where the emissions are greatest. So the reason why shallow waters have larger bubble emissions normally is pure physics. So there is less pressure. So the dissolution rate is lower there, so production, so it can be so they can be released more easily due to atmospheric pressure. And of course, if you go to 10 meters, you're at 2 atmospheres already, and it's harder to be released. So that's my understanding of the reasons why emissions are greater in shallow waters. Also, maybe a bit of turbulence stirring up the sediment slightly, or yeah. But the differences between the different ends of the lake, I'm not entirely sure yet. We have organic content for the sediment under every chamber, which we will use to try and explain it. We also have sediment texture and consistency. They could be some explanation. It could also be that there's where the direction of the prevailing wind that
similar in their effects
makes all the algae back up at one end of the lake. Always a sun coming in one of my talks. At least he didn't sit on my lap this time. So yeah, we're not sure yet. Is the short way of saying what I just said in a long way, but we hope to find out.
Hi, Tom. Thanks for the talk and also for the summary of all the presentations in the session. The results are very nice, and I also wonder about other factors that might affect the pattern in methane. So you don't see a potential connection with biota. It's like a fish, benthic fish in this lake producing or facilitating a release of bubbles from the sediment, or that could be indirectly affected by the oxygen application and so on.
Yep, could be in the margins. We see very big emissions in the margins in the mixed phase. So when the lake is oxygenated at the bottom, the emissions follow the more traditional pattern of larger emissions in the margins. I mean, the work on benthic fish has actually found it overall reduces methane bubble emissions because it aerates the sediment. So it kind of works both ways. It's not that their winnowing behavior causes bubble release. So it goes two ways. I guess fish would be largely excluded from the sites of the largest emissions during stratification, because it's really anoxic. Fish would be avoiding those environments. So the places where, yeah, it could be that, could be an indirect contributor in that they are not stirring up the sediment, so emissions can be larger. That would fit with the literature. That's a good point. So Nesta, you had a question too.
Hi, Thomas. Nice to see you again. I hope you are very well.
Thank you.
Beautiful, beautiful contribution. I have the same question about the main driver that explained the spatial heterogeneity in the transect that you consider. But it's the same question that Mariana indicated previously. In this sense, I want to go to the second question. If we want to estimate the emission or the sink at lake level or at ecosystem level, this heterogeneity is a kind of problem because demand an incredible huge effort in terms of sampling. In this sense, some estimation like a phone number that probably is far away of the reality. What do you think?
Yeah, I saw your question on the comments. I saw your question that's similar on the question section, and yeah, when you look at the variability of an individual point, it's a bit scary, isn't it? We need to find a better way of measuring. I don't know what that is right now, but hopefully we can find one. And also, this 40 chambers, I think, is overkill. I think we can do it with fewer, and this will give us some clue as to how many we need. We can do some nice analysis and randomly resample all the chambers and increase the numbers and see which gives us the closest estimate how many we would need to use and what depth they can go at. Because it's not realistic. This is a one-off possibility to do this project like this, and I think we should take advantage of it and see how often. I think the time is the more important than number. I think not 4, but maybe 10 across the whole lake would do something close to this. But you need it every 2 weeks. You can't. The time is really key here. That is my guess at the moment, but we'll find out. Sorry, then Sophia, you had a question.
Yes, hello, Thomas. Very nice
Hello.
presentation. At the end of your talk, you say that you want to see what will happen with these pulses during the mixing when the submerged macrophytes appear. And I was wondering, like, if you had to guess what will be the effect of these submerged macrophytes, if you think that maybe they will have, like, I don't know, a physical effect over this, and they will, like, retain this methane, or just if you had to guess, what do you think?
Yeah, there's nothing that. Yeah, we'll find out one way or another, isn't it? Hopefully macrophytes will grow this coming year as the lake has been biomanipulated and most of the zooplanktivorous fish removed, and it's already looking much better now, much clearer than it was this time last year. My guess is that methane bubble emissions will be dramatically reduced, which is what we see in our experimental treatment, and what I've also seen in a few real lakes where we've put chambers over plants and over the open water, even within the same lake. In areas where you have plants, you have almost no bubble emissions, and in areas where you have no plants of the same lake, you get a lot more bubble emissions. So my guess, there will be a dramatic reduction. As to the degree to which it reduces the effects of stratification, it's hard to say. It depends on the year and the temperature, and we'll see. But I would strongly higher. Yeah, I'm pretty certain the reduction, the emissions will be an order of magnitude less. But yeah, we'll find out. So sorry, I'm taking up too much time here on my talk. But one last question to Annelies, who's had her hand up for a while.
Yeah, thanks, Tom. Very nice talk. I think you mentioned that you didn't really see a temperature effect on the methanogens. And I was wondering if this could have been confounded by temperature effects on the methane oxidizing community, or maybe an oxygen, like a temperature-dependent oxygen effect even on the methane oxidizing community. Do you have any idea about this?
Yeah, I mean, we don't know what was happening in the microbial community, because we haven't sampled it, but we know there was a huge increase in methane production with I think it was 10 million% saturation in the bottom waters. So I think methane oxidation basically stopped. And lake is maybe a really extreme example because it's nitrogen limited, so there's very little nitrate. So it's really down that end of it's heading towards fermentation conditions really quickly. And so 3 weeks without oxygen, it was really producing a lot of methane, and there must have been very little oxidation. So did we increase production? Almost certainly. Was the production increased? And oxidation was also massively decreased. And how quickly can they come back when the bottom is oxic? How quickly can how long can methane oxidizers survive in an anoxic environment? How quickly do they come back? But I reckon the greatest potential for oxidation would have been at the redoxcline or the thermocline, where you would have had 10,000 ppm methane meeting, I think it was around 200 in the top waters at that time. So there's a lot of methane and a lot of oxygen, so they should be there. And then maybe we should look for them next year. But I guess oxidation was just totally wiped out for those 3 weeks.
So the zone was just too narrow for them to really be a filter.
I guess so, yeah. And they can't, and they're not on the sediment surface anymore because it's totally, or they're not active, at least. They probably are there, but they're not active.
So, and then I was wondering, if the lake is deeper, would you expect the filter to still work, like to still function in water column?
But yeah, they need, I mean, what I'd really like to have looked at is how much methane production was actually going on in the water, because it was anoxic for a long time, for several weeks. Would the methane production move into the water column or not, or stay in the sediment where we traditionally think it happens? The anoxic version, at least. Yeah, but I can't, and they're really interesting questions, Annalise, but I can't really answer them. So you need to come and answer them for us one time.
Sure.
Okay, I think we should move on to the next presentation now. And this is by Chiara. Am I sharing? Not yet, sorry. Too many buttons to press. This is by Chiara Esposito, who's working as a PhD student with us in Succobo. And she gave a nice presentation on patterns and processes of methane production and oxidation in the different habitats of our experimental system. So we have these patterns as a background of bigger emissions in some tanks than others, and we want to know who's making, or Chiara's study is interested in looking at who's making the methane and who's consuming it. And here we focus mostly on methane consumption. In this presentation, at least. So we have the different parts of the ecosystem that produce and consume methane. It's mostly produced in sediments and probably in our systems exclusively produced, although there is increasing evidence of oxic methane production, but that's a slightly different process. So, and then we look at all the different habitats where methane could be consumed: on the walls of the tanks, the periphyton, in the ceston, on the macrophytes, in the filamentous algae. And there's a lot of work in this. So we measure ebullition and dissolved concentrations. So there's a lot of different work you would have seen in the presentation, a lot of incubation experiments. And to summarize really the presentation is that we see quite a lot of methane consumption going on plant surfaces. And we have hints of a difference between the different species. When you have two species in the same tank, there's often a bit of a difference at low nutrients. At the bottom is the roots and at top is the above ground surface. So we do see some differences. We don't have enough samples to really test for significant difference. So we see relatively even consumption of methane on the plant surfaces across the tanks, and then we see a lot higher consumption in the high nutrient tanks. And there is much higher consumption on the least surfaces than that associated with the roots. So we can now stop sharing again. And now we can have questions for Chiara, and Nestor has his hand up unless it's an old hand. Over to you, Nestor.
Very nice contribution. The graph that actually showed Tom indicate very big difference between Elodea canadiensis and Potamogeton crispus. Which are the main possible explanation for that, Chiara?
Yeah, thank you for the question. We actually don't know yet, but we are just we are working right now on molecular biology analysis. So we want to see which kind of communities of methane oxidizer bacteria are in both species. And we think that the switch in communities might indicate and help us to understand the differences in methane oxidation rates.
Hi, it's me. I put a question in the web page. It's more about the further steps that you're. I think it's the same experiment that first Tom showed, and he measured the things in the sediment. And here in your further steps, you say that. What I was wondering, what do you mean by different habitats from each tank, like to define the bacteria composition? So which habitats are you thinking?
Yeah, exactly. So it is a bit of a follow-up of Tom's experiment, but he focused on the sediment, and we wanted to have kind of a total balance of where methane is produced and consumed in a shallow lake or in our mesocosms. So we sampled the sediment, but also the other habitats that in our case are the microphytes, the periphyton, so the walls of the mesocosms, the filamentous algae, the water column, and yeah, so these are the different habitats. And we not only measure the consumption rates in the sediment, but through all the habitats. And yeah, the last step that I put in the presentation, it was just the thing I said before, that we would like to see which methane oxidizer bacteria are in the different habitats. And this we think could help us to explain the differences in methane oxidation rates. Yeah.
Thank
Did
you.
I answer?
Yes. Yeah, I was wondering, is this really novel? Because I guess a lot of people is focused on the sediment, right? For this, or not really? So will this be?
Yeah, exactly. There's a lot of literature about sediment, but not so many about the other habitats. In particular, what was interesting was also that we found some high consumption rates in the submerged filamentous algae, for example, and in the periphyton. And there are not so many studies about this habitat. So yeah, well, let's see. We are curious about the new results that we are going to process. Yeah.
And this would probably all will be nicely linked with the real manipulation and eventually mitigation strategies, right, of the second home big lake of the adding macrophytes. So it's all like trying to prove that the macrophytes help to reduce.
Yeah, or at least there could be habitats for methane oxidizer.
Okay.
Yeah.
Thank you.
Yeah, you're welcome.
Okay. Any more questions? Sure.
Yes.
Oh yeah, sorry, I
You,
missed the hands.
Chiara. I'm sorry. So we are too many in the session. Thank you, Chiara. I wonder why you think the bacteria communities will differ. Do you think it's the microhabitat within the microhabitat? So it's the abiotic template, or there might be different grazers on the bacteria due to their habitat created by the sediment or the different macrophyte species that can lead to different bacteria community. So do you have any idea what would be the process more inside the microhabitat that could lead to such differences?
Yeah, that's really, that's a really interesting question. I don't know, I'm not sure about the answer, but I think what we would like to focus on is also the influence of different temperatures and different nutrients in the different oxidation rates, and so in the different microbial compositions. So yeah, we expect differences due to temperature and due to nutrient levels in the tanks. Yeah, but I think that also the composition of each mesocosm, so the presence of macrophytes or not, the presence of filamentous algae can then lead to differences in microbial communities.
I think it's very interesting that that all the works you've done in Aarhus are showing all these different scales of factors affecting. So you have the within lake, so you have the depths, you have time, you have the habitat, the macrophyte and non-macrophyte, and then you within the microcosms you're going into even finer view or zoom. You're zooming in to go even smaller and smaller scale processes. So in the end of the day, what do we do? Where do we measure? Where do we go and check for the processes? So we have all these different scales and time and space acting at the same time. And that, of course, imposes, it's impossible to measure everything. And also, it's would be interesting to try and reach patterns. So where are the most important processes happening inside lakes and inside the different habitats? Or what time is the best to capture the most important pattern in terms of emissions? So I don't expect you to have the answers, but I think it's very nice and see all your findings together, and I end up with more questions than certainties.
Yeah.
Thank you again.
I think with the plants difference is really hard to say. It's a really initial study, and it could be that to do with there's so many routes of methane out. Is it coming up through the plant and leaking out? Is it just in the water? Could it be that Potamogeton crispus, which has better roots than Elodea, actually transports more methane internally, and so provides more methane for the oxidizers? There's all sorts of how far. You're right. How far we have to stop at some point. We can't go to every single scale. So this is an initial.
I didn't mean to say.
Okay, we have to stop at some point here. Thanks very much, Chiara, and thanks everyone for their questions so far. We'll move on to the next. Oh, again, I'm on the wrong place. So now we have, no, that one. Can you see the right slide? It's, yeah. So this is the presentation by Gregorio Lopez Morea. I hope you got your name right. And it's a impressive mesocosm investigation looking at kind of a multi-stressor approach to looking at the effects of a variety of stresses, including various toxins on metabolism in shallow lake ecosystems. And there's a lot of results here, so I can't present them all, but we can have time for questions. And it's just an outline of how runoff from land, including a number of pesticides, insecticides, and nitrates, crossed with temperature, so cool tanks and warm tanks. So this is a gradient study, a gradient of the stresses, as opposed to like at Lemming we have treatments and replicates. This is a different approach to mesocosm research, which is also really interesting and a good approach. So they have the same gradient of stresses at cool and warm tanks, and then looked at metabolic rates across these tanks using diurnal variation in oxygen. So there was too much detail on this for me to really summarize well here, and I'm sorry about that, but I think that was that for now. So we can again shift the questions to Gregorio, and there you are.
Yeah, hello everyone.
So Mariana has her hand up, unless it's an old hand. Old hand. So, excuse my pronunciation, Gulce. Gulce.
Yes. So, hi, Gregoria. It was a really nice presentation and
thank you.
Great mesocosm design, I think. We're also planning a mesocosm experiment where we will do the heating, and we also do it in the water because our system is in the water. That's why I was wondering some information about the mesocosm system. Like, did you use any insulation in the heated treatments, or did you just use the water in terms of insulation?
Yeah, okay. So I don't know if you saw the photo that I included in the slides, but our mesocosms were all inside a bigger pool. It was like a bigger basin in which we had all our mesocosms. And what we did was to heat up this water outside the mesocosms, trying to get the water inside the mesocosms to be as uniformly warmed as possible. Actually, I don't know the very technical details about the heating system. I would suggest you to contact some other people in our team. You can contact me via email, and then I can forward it to the right people, the ones who developed this heating system. But yeah, what we did was to heat up these basins, the bigger basins, and then we relied on that to keep the mesocosms warmer. And as you can see also in the plot where I show the average temperature for all the mesocosms, both in the colder and the warmer treatments, we managed to keep the warmer mesocosms around 3 degrees higher throughout the entire experiment. And that was really nice to see that the heating system worked really well.
Was there an automation in the heating system, or did you?
Yeah.
Yeah. Okay.
Yeah, it was automatic, and we were also measuring evaporation from this basin and replenishing the water in the mesocosms regularly. So because this heating led to higher evaporation, of course, and yeah.
Okay, thank you.
Okay, Tom has
thanks.
a question. No, sorry, Lana first, then Tom, and then I saw Erik's hand up.
Okay, thank you. Hi, Gregorio.
Hello.
I enjoyed your talk a lot. I just have a question. If I understand correctly, your conclusion was that your phytoplankton and macrophytes were quite tolerant to the pollution. And I was wondering, did you not notice any blooms or toxic cyanobacteria or anything else? And I was just wondering, you say that the mesocosm was tolerant to the pollution, but what about the entire ecosystem, like the other levels, zooplankton or anything else that was in your tank, or was it in your tank? How did they react to the pollution? Because it seems to me that your conclusion is like, oh, they can tolerate it, but
yeah.
Like, autotrophic part of your food web, but what about the other ones? That was my question.
Yeah, so in this presentation, I actually focused on the primary producers because what I did was to analyze the metabolic rates, and I focused particularly in primary productivity, right? But there are other presentations in which they are like also from the ClimShift project that you can find in the program of the conference. And also one of my last slides, I'm showing all the presentations from this project. Where they are also checking the other compartments, so the zooplankton and all the other compartments, let's say. So what I did here was just to draw a preliminary conclusion that is pretty much all we can conclude so far, because we are still working on several other analyses and looking into the results of microcosm experiments and developing mathematical models to try to understand better the dynamics that we observe in the different experiments. And the mesocosm experiment was particularly confusing to look at, because we had these very different responses from different mesocosms. And one of the common aspects that we can see in all of them is precisely that the primary producers seemed to find a way to thrive anyway, no matter the toxicants, and in all temperature conditions, they sort of adapt to keep the productivity of the system high. And yeah, this hints at the resilience of the mesocosms that we worked with, I guess. So yeah, so far we cannot say much more about this. Yeah.
All right. Yeah, they tend to be the winners in all our treatments, right? The algae.
It's either phytoplankton or the macrophytes that are dominating in the end. Yeah.
Yes. And can I ask an additional question? What were the specific toxicants that you use, like what kind of chemicals were pesticides or
I'm sorry to interrupt here. Can we take these technical details later, because we're going to run out of time for the other people's presentations? I'm really sorry about that. If you could ask questions online or mail. We'll take one more question from Tom and then from Erik, as you had your hands up, and then we'll move to the next talk.
Yeah, I had more of a general thing, which is what do you like? I find it interesting to have these different mesocosm designs where it's either a treatment or a gradient design. And what do we think is better? I thought it would be a nice short discussion point, and maybe you can give your opinion on that. Well, I think there's some, yeah.
Yeah, I think in future experiments, I would go for a setup that includes more replicates, because like I said, we observed very diverging behaviors or responses in our mesocosm. So it's really hard to generalize the response when you do not have these replicates. So I think the gradient approach is quite powerful, but I would definitely recommend to include replicates, just to be sure that what you're observing is the actual response of the system in a general way, and not just like something that perhaps happened and drove the system towards a particular behavior. Yeah, but I agree that this is a really important discussion that needs to happen at some point. Yeah.
Okay, Erik, I'm sure you have a very quick question.
Nice multi-stressor experiment. It's very nice to see these kinds of experiments. I have a problem with your respiration and production calculations. I see that in many of your slides you presented that you have 0 respiration for a long time period. And if I got 0 respiration, I will die. And I think most of the others will also. So it's, I think there's some calculation problem you're dealing with here, and it could have very big effect on your final net production discussion, because how can you get 0 for so long time? Is that because you get negative values and then you say it's 0? Or how do you reach this 0 values? Because it's not normal with calculating from oxygen. And when I see your oxygen data, this should also, you have a diurnal variation that should give some production and respiration over the time period. So there's something that you have to look at, I think. It's not, but I know it, you are only in the process of analyzing the data, but this is very strange to me, for me.
Yeah, we actually looked at that also like within the group, and we find it also very strange that we have this very low. It's not really 0, but it's a very low value of respiration. So we are still trying to understand this better. But yes, I agree that this is something that we should definitely look at.
It could be something with the modeling calculations that you are.
Yeah, my guess it's related to the exchange with the atmosphere. So probably there is something that I need to recalibrate there. But I've tried different versions of the model, and I always end up getting this. I even tried with an R package with Stan and also with JAX in MATLAB, and yeah, I end up having the same results. So yeah, perhaps someone is more knowledgeable on this. I could get some pointers from them. Yeah, definitely. But thank you for the comment, Erik.
Okay, thank you very much. I just add that that piston rate gas exchange is a world of pain to enter. So good luck with that.
Thanks.
I'll move on quickly to the next topic. Yes. So this was sorry. Not correct one, I think. So having some technical difficulties here. There we go. Start again. So the next talk is got too many screens open. Meredith's nice presentation. I hope you can see the screen, and I'll summarize this quickly. This is a very nice study, which very interesting topic of how common it is for very small water bodies to stratify. And what Meredith presented was this really nice results of 3 different kind of classifications of stratification. So often, mixes often, intermediate, and really mixes, and then what the drivers of those are in terms of depth and exposure and size. So showing that small water bodies actually stratify a lot more than has been traditionally thought, and this clearly has implications for all sorts of processes, not least greenhouse gas emissions, as we showed. This links very nicely to our work from Ormstrup that we presented earlier. So you need to upscale that sort of information. You really need this sort of information. It's a very thorough study of those of that phenomena. So I think we also have time for a little discussion of that. So is any questions for Meredith?
It's not a question what I have, Meredith, but it's a beautiful work you did, and the presentation was extremely nice. And I think it's giving a strong foundation to a change in, I wouldn't say paradigm, but more or less about ponds and how their physical behavior can have extremely relevant effects in their biology and functioning. So it was just a comment to congratulate you on your work. And I think it's going to be very, very cited, given the implications of these nice findings.
Yeah, it was really fun to see Tom's work. So thank you. It was exciting to see Tom's work and sort of where we see this going with how these different mixing regimes could have very different implications for the greenhouse gases. And it's been a really fun project through Glion. So if any of you saw that and were excited, we're wrapping up 4 projects right now, and we're trying out some new ideas this summer, and we're planning to launch some new ponding projects next year. So we would love to have a broader representation. As you could see, it was North America and Europe. And there's a lot of people on the call from other areas of the world, and we would love to collaborate. Shameless plug.
Yeah, we'd be, it's great to see this study. Is actually, we have a pond project starting at the moment, and I was insisting that everyone put thermistors in their ponds. So, but you beat us to it. But at least we'll be able to drag it too.
Yeah, no,
But was three enough? Is my question actually.
It is for this like first course categorization of whether they're mixed or stratified. But we're working on a metabolism project right now. And some collaborators didn't put thermistors in, some put 2 in. And to get at that Z mix question, we're finding that more thermistors would really be better. And a thermistor above the DO sensor is pretty critical, which we didn't think through at the beginning of the project. So one of the take-home messages from the data analysis on the metabolism project is that more thermistors are better. If you've got a small system, wind doesn't seem to matter. So put the money towards the thermistors as opposed to measuring on-site wind, versus the bigger sites, wind matters and thermistors don't matter as much. So it's sort of when we're thinking about equipment and we're trying to get lots of people to do these measurements around the world, there's definitely a priority of what we should measure versus what's not going to matter as much. But if you had lots of thermistors, I'd say probably 4 to 6 would be better than 3, but you can get away with it.
Thank you. So, unfortunately, we don't have unlimited time, so we have to move on to the next presentation, which, maybe I should have just talked all at once. It would have stopped all this clicking, but I think it's nice to stop in between. So this was a nice presentation on a topic which fits with some of the things we've already discussed by Sofia Balinja, greenhouse gas dynamics on shallow Pamplean plain lakes with alternative equilibria states. And just to summarize, that's not fitting very well, but it's to explore the difference in what we've been discussing about carbon-based greenhouse gas emissions from lakes, ponds, Pamplean ponds, with abundant submerged macrophytes and without, so in the 2 alternative stable states. And what I liked about I liked a few things about this study, but one of them is the real thoroughness of measurement. So dissolved concentration and then chambers and then bubble emission traps too. So you can really see they really tried to get every angle of emission. And then the summary of the fluxes, there's quite a lot to summarize here, but the difference between clear and turbid sites was that we got more influx of CO2 in turbid sites in spring and summer, in clear water sites, sorry, and more in turbid sites later on in the summer. But there wasn't very much difference in methane emission between the two sites, which I think we can discuss a little bit now. So I'll stop the share again. So Sophia, thanks very much for a very nice presentation. If anyone has any questions, I open the floor.
Thank you.
Nestor, please.
Sophia, nice to meet you. Congratulations for the presentation. Actually, I sent a couple of comments to you, but I want to continue. You can comment some results about the difference between ceratophyllum and myriophyllum in terms of the emission of methane and CO2?
Actually, for the study we did, I couldn't differentiate them because when you see them in the clear lakes, they are forming like these mats. They are all together, so it's pretty difficult to disentangle the effect of these both submerged macrophytes. But what I tell you is that is related with the question you put in the questions and answers, is that in the clear lakes, the submerged macrophytes are covering all of the bottom of the lake. They form like real grasslands. So when I do a chamber, like in the open water, or when I get samples for headspace to measure the gases dissolved in the water, this is always including the submerged macrophytes. There's no such area of open water without them. So I couldn't tell you exactly the difference between both of them, but they are both included.
Okay.
Okay, thank you. So Meredith is next on my list.
Yeah, I thought this was really fascinating too. I'm a little surprised that the turbid waters didn't have greater greenhouse gas emissions than the clear water, and I don't know if you were expecting that too and were surprised. And I'm wondering if that might relate to benthic dissolved oxygen, if that was similar or different among the lake types. I might expect stratification to differ, but again, you're in a more open landscape, so maybe they're just completely mixing. So I'd love any more insight on that.
Yeah, actually, it was very surprising for me, a lot of results that we got, because I was expecting to have more differences between the states, and you finally see like, okay, overall, it's like they are both emitting more or less the same. So regarding stratification, for example, in oxygen, we can find a different stratification. Like the turbid lakes, if you do a profile, you will find similar oxygen only in the water column. But then in clear lakes, you can find differences, but we didn't see this in temperature. So to combine all of these and to make an idea of what will happen, it's a bit difficult because there are a lot of factors working. But in the overall, they tend to emit the same. Also, we have to remember that in the clear lakes, we have the plants, and maybe they are also having some kind of physical effect. So I don't know if I'm answering your question correctly, but I would have expected also differences between the regimes.
Yeah, thank you.
Thank you.
I have a question related to that. Maybe I missed it in the talk, but did you talk about what species of plants there were present, and also the chlorophyll A values of the two different tanks?
Yeah.
Oh, sorry,
the submerged.
Lakes.
Yeah, lakes, ponds. Yeah, the submerged macrophytes were ceratophyllum and myriophyllum. which are two submerged macrophytes that we have here in the Pampean Plain. And then the levels of chlorophyll A really differ between the clear and turbid lakes. In clear lakes, we have like 5 micrograms per liter, whereas in the turbid lakes, we have found up to 80 micrograms per liter. So it's like an order of magnitude of difference. Yeah, the clear lakes are really clear. You can see the bottom.
I would just have two small comments on that. In our experiment, we're dealing with massive differences in chlorophyll A. So in our experiments, they can be over 500 micrograms per liter in the turbid tanks sometimes. Of course, they're lower a lot of the time. And also, anecdotally, I know of a study in Germany where Stratification demersum was actually associated with rather large methane emissions. Yeah, which also really surprised me. So it could be that plant identity is actually important, and Stratification can hang around in a relatively unhealthy, dark, non-photosynthesizing state for quite a long time. So that might be of interest. So is there any more questions before we move on? No? Okay. Thanks again, Sophia. So next, we move to Maria Caldero Pascual's presentation, which was, and I think this is a picture of Erik here on next to some mesocosm experiments, if I'm not mistaken. Gets everywhere, even though he's not on the author list here.
No, it's not him though.
Oh, shame. And it's a really nice, we'll hear a lot about this experiment in the next talks, which is an experiment looking at the effects of adding dissolved organic carbon of various sources. 3 different types here. One, this human feed. So a pulse experiment adding human feed, which is high color, low DOC actually, and recalcitrant DOC treatment, and then an alder leaf leachate, which is much higher DOC and more labile DOC, and then a combined effect of the two to these different mesocosms in Turkey with a nice, really nicely run experiment, 9-day acclimation, and then the pulse, and then looking at the effect. And the study found a quite clear difference in the type of DOC quality. I think this is the Parafac result, and looked at the implications for that on the food quality of some of the algae, and then looked at overall treatment effects on a variety of things: carbon quality, DOC quality, cestin quality, nutrient quality. And I won't talk about this too much, but it was basically the labile forms and the combination of the labile and human forms which had the greatest effect. So now the floor is open to questions for Maria. And I think Meredith, that might be an old hand, but if it's a question, go ahead. So no questions?
So maybe I can ask a question. Yeah, really interesting, this work with different carbon sources. I have a student who's going to start a project on that relating to methane emissions. And I saw that in this experiment, you used recalcitrant and leaf-based carbon. But how do you think if you would use elk, and that's not only for you, but also for Brucet, how do you think if you take, for example, carbon from macrophytes or carbon from algae, how would that affect the quality of the carbon and the nutrients compared to the leaf and the recalcitrant carbon?
You mean when you mean by algae, you mean like having a really, like taking the DOC from a really high altitude trough tank, or like
I know an experiment where they had algae. I think it was probably filaments algae, and they how do you say that, dry freeze, and that's the, yeah, okay, they dry froze it and then made it into like particles, and they added that to look at methane production. And you could also add that rather than leaves, of course, or something like that. Do you think that would give a very different result here?
I guess, Brucet, if you want to add something, but here at this point, if we are talking about bacteria, it would be a really tasty DOC because it would be autochthonous dissolved organic carbon, but also thinking about zooplankton then, that they would directly maybe graze on the cell, not on the excreted carbon. Maybe then I would have to say that it depends on the type of algae. If they are maybe, yeah, depending on of the zooplankton community and the type of algae that you add. So here the point was, and you brought it up here, it's very important to characterize what you are adding to understand what is going on in the community, because it will change depending on what you add. Even though all it's just additions of alloctonous organic matter, it really, your results can really depend on the characterization of this type of organic matter.
I think it also depends on the question. The reason why we use the alder tree leaves is that we wanted to test the impact of local DOC that's coming from surface runoff from the catchment. That's why we used alder tree leaves. So if your question is...
I can also add, we ran an experiment a bit like this as part of Aquacosm 2, and I was very interested to use algal sources of DOC to look at kind of within lake effects as opposed to without lake effects on methane production and general ecosystem biogeochemistry. And you have to do flask experiments if you want to use algal DOC because it's so hard to get enough, you know. We had to make you have to make a 4,000 microgram per liter solution of DOC to get enough to make a 2 milligram per liter DOC mix in your 2,000 liter tanks. So it's the practicalities of an experiment and in a mesocosm experiment. I think it would be all near impossible to get enough algal DOC to add. So its practicalities is hard.
Yeah, we plan on using aquaria, so it's about this size. So that could, that sounds like it might still be feasible. And then we have to find a place with a lot of algae, probably drainage ditches or something like that.
But the next experiment we are going to do at Meiji, I think, is to use dog shit, because there are more dogs at Meiji campus than there are students. So it's using some dog shit. So you're welcome to come and eat a dog here.
That's a really shit experiment, Erik.
But it's the only one in the world.
Okay, moving swiftly on. But are there any more questions to this? We are running out of time, so I'll take the next one, which is that's a hard name for me to pronounce. Is it Thais? Sounds a bit like a Danish name, but it's rewetting. Looking at the effects of rewetting of marginal sediments from a reservoir on sediments with and without agricultural crops. And you can see where the sampling was done with crops and without crops, and then an incubation afterwards, and then measurements of the greenhouse gas emissions from both CO2 and CH4 from the cores with them that had crops and hadn't had crops before. So I can now open the floor to questions. To Thais. Maybe my first question is, how do you pronounce your name?
Thais.
Thais, okay. Thank you.
It's a Brazilian name.
Nestor.
Hi, Thais. Beautiful contribution. It is possible that you explain the pattern of dry and rainy period in around Natal, and also for several contributions in the case of this Congress. I can assume that, for example, some dry periods last several years. I'm okay.
Yeah. Yeah. This region has faced a prolonged drought that began in 2012 and lasted until 2019. So it was a 7 years period of drought. And did you ask anything else?
It's okay. It's okay. It's only to understand the big picture, what happened around the Natal shallow lakes. Thanks, Thais.
Any more questions? I have one, Thais. The nice experiment and shows nice, clear results. But of course, it took place in the lab. How do you think it? How do you think it will relate to what would happen in the reservoir when it actually the water level raises and the land floods? Do you think there would be similar results?
So when we carried out the experiment, we knew that there were some limitations in the adopted experimental design, because our experimental design aimed at blocking photosynthesis because of the dark period. And also the experimental design aimed to block some organic matter that could be present in the water, in the reservoir water, because we put in the cars distilled water. So the distilled water doesn't have organic matter, right? So we have these two, maybe I can say limitations about the experiment setup that we made, and these two things that may have some I don't know how to say, some limitations in the results.
Okay, thank you, and thanks for your presentation. Okay,
Thank you.
I have a little time left for Gulce's presentation, and here we go. It was a very nice presentation on the response of the microbial loop to this carbon pulse experiment that was carried out in Turkey. I put it on play. To look at how climate change driven DOC disturbance might affect the food web, but or the microbial loop might affect things via the algae. And it was a really nice study with these 4 different treatments again, the same with the human feed, the alder leaf, and the mix of human feed and DOC. And I think I was quite brave putting this slide up, because then I have to try and explain it, and it's and I'm not the author, but it's really there's a core looking at bottom-up effects of the DOC and where they were significant. So there was a significant effect on the ciliate community with the leaf leachate, and a significant effect on the zooplankton with the mixed treatment, and a significant effect on bacteria by the different DOC treatments, 2 of them, only the mixed treatment and the label treatment. What I really liked is this bacteriary treatment dependent response, where the ciliates got a larger portion of bacteriivores. That was very neat. So we saw kind of food web feedbacks. So I'll stop sharing and we go to the last round of questions for Gulce. So Tom has a question.
Yeah, Gulce, a very nice presentation. It was very clear. I thought it was very interesting. Also, the part that Tom said. And one thing I wanted, I remember there was this part of the effect on the microbial loop, and the effect was much larger if you had the mixed DOC than only the labile DOC. So why do you think that is? What does the recalcitrant DOC add that makes this effect go up? Rather, because if you only added recalcitrant DOC, nothing happened, but somehow it interacted with the label DOC.
So first of all, with the recalcitrant DOC, not as much as we anticipated, but it comes, shading effect, and this increases the impact on the bacteria with a faster response. And another one is, besides from this shading effect, there is also recalcitrant DOC. I know that it's recalcitrant, but together with the mixed with the labile DOC, it has also providing some nutrients. So the impact is, synergistic in that sense. Maybe Maria can add more onto this, since she also investigated.
Oh. Yeah, maybe. Yeah, I think you kind of answered. I was also wanted to add the bit of the quantity. It's always about quality and quantity, and the combine it, in reality, has slightly more quantity, but also double light or shading effect. And this could be potential reasons. We don't have phytoplankton or like the autotrophic bit to maybe clarify these advantages over, But I would say, yes, as Kulsha said, the synergistic effects in this case regarding these other aspects.
Thank you for the question.
We have time for one more. Well, we don't have any more time, but we can have one more question if someone has any. No hands up. Okay, then I would like to thank everyone for coming to the session and everyone who went to the trouble of, and sometimes not inconsiderable, of recording a talk and uploading it. And thanks for being here and coming with your questions. It was really interesting and some really nice talks. And I really hope to see you all in person next time. So, bye-bye.
Thank you.
Bye.
Thank
you. Bye.
Bye-bye.
Bye, thank you.